All right, so we are now recording. Thank you very much, Steve, for giving a Build a Cell seminar, and it's all yours.
Okay, let me see if I can now share the screen. And what I'm going to try to do here is, I will try two sharing strategies, but I think the easiest thing to share is, I can put it on slideshow, but then I have to share a different window. So let me try slideshow. So what do you see on your screen?
Nothing right now. You're not sharing anything.
Okay, good. And that's a problem here is I don't seem to be, let's try this one.
Now it's perfect.
You see the slide?
Yep.
You see the meeting is recorded on the slide?
Yep.
Okay, now I've removed it. I'm hoping I don't see it. What do you see now?
I see just your slide, and there's a dictation mode little window.
Oh, great. Let's get rid of that. Okay, excellent.
Perfect.
So how is everybody doing? Who here has had the pandemic? Who has got COVID? If you have, raise your hand. Does anybody know how to raise their hand? No, I guess no one got the coronavirus. That's good. I interview everybody now I talk to who has had it. So we have about 200 case studies now. But this is not the topic for today. The topic for today is biotechnology and artificial cells floating in fluorinated media having the same density as water. And as you know from your work with Kate, there are actually a couple of reasons to make artificial cells. One is a historical one, right, related to how life originated. And it's presumed that compartmentalization is important because altruism is contrary to Darwinian evolution, as is parasitism. And so the idea is that if you're going to do Darwinian evolution at some molecular level, some kind of compartmentalization is required. And of course, lots and lots of people therefore try to make sort of biologically relevant compartments that are likely to be made of materials that were available on early Earth. Now, this is absolutely, we do a lot of work on the origins of life. This talk will have absolutely nothing to do with this. What I'm going to do is focus on the technological applications, which arise in sort of the same reason, that is you want to connect phenotype with genotype, but here you're trying to do directed evolution of enzymes. And 20 years ago, Tawfik and Griffith, we thought had solved the problems, actually more than 20 years ago, using water and oil emulsions, which I'll talk about more in a minute. And then a former student of mine named Phil Holliger is now at Cambridge, England, published some papers in the roughly 2003. Gadesi et al. Is the first reference, where they actually evolved polymerases in a compartmentalized self-replication architecture. And there, you put stuff into the droplet, and if it's a polymerase you're trying to evolve, you lyse the cells that are holding each one a different component of a library of polymerases. In the cell is the gene. Therefore, in that droplet that you've lysed the cell into, you have both the polymerase that has been expressed from a plasmid, hence the phenotype is there with the genotype, and if the polymerase is able to replicate its own plasmid, then you get a lot of the polymerase copies of the polymerase gene that replicates itself, and this is the self-replication. Now, we are desperate, just for the record, we are desperate to be able to, engineer polymerases that accept an expanded genetic alphabet. For those of you who have not heard a talk from mine in the area, here's it briefly with color schemes. As you all know, Watson-Crick pairs follow two rules of complementarity. You have small things on the left pairing with big things on the right, and that's size complementarity is what makes the rungs of the DNA ladder roughly the same size and therefore interchangeable. And then you have hydrogen bond donors and acceptors. Here the donors are in red, the acceptors are in blue, that are matched. So cytosine is a small thing with the hydrogen bond donor acceptor matching the acceptor donor, blue, red on the big thing. And I actually have put here aminoadenine instead of adenine. As you may know, in the original DNA that you likely emerged on Earth, and also what you have in your body, you actually don't have the third hydrogen bonding group at the bottom of adenine. That is, you don't adenine is incomplete in its hydrogen bonding potential. However, a couple of weeks ago, you may have seen Phil Morlaer and various other people actually found cyanobacteria that use this. So I now use this as a natural way of showing complete Watson-Crick pairing. So there is an acceptor, donor, acceptor thymine pairing with a donor, acceptor, donor on aminoadenine. But a number of years ago, we figured out that if you shuffle the hydrogen bonding donors and acceptors, you can get orthogonal pairs which have different patterns of hydrogen bonding. So the one that we will talk about a lot is the one with the pyrimidine PY donor acceptor, that is red blue hydrogen bonding pattern on the small thing, and that's in the middle on the right hand side, and that pairs with the larger structure. And so these have various names. So that pair is the ZP pair, above it is the BJ pair. Z comes from Zuni, and Pin, she is the person, Zuni is the person who made the guy on the left, and Pin is the one who developed the P base. This is not International Union of Pure and Applied Chemistry nomenclature, but you'll hear me referring a lot to PC in this. So there's a 12-letter genetic alphabet, and we very much want to have replication of it with polymerases that do so. And so, since Phil Holliger was a former student of mine, and he basically solved the problem, so what we thought we would do is just do a compartmentalized self-replication in his droplets, where we amplify the gene for the polymerase, which is that red line, with an external primer that contains the funny base P, that's the magenta things in the external tags. Once you do the first cycle of PCR, the amplicon has tags with the funny base P, the component of the Aegis system, which we're going to expect you to have memorized by the end of this talk. It's the one in the middle on the right. And then, of course, once the tagged polymerases, once the tags are added to the amplicons, the PCR is carried by primers that are external to the gene that contain 4 Ps. And so if the polymerase doesn't incorporate Z opposite P, it doesn't amplify its own gene, and we're supposed to be able to therefore select for these. That's the story. And of course, there is sort of the schematic now with the compartment itself. So basically, you put E. coli with a library of polymerases into one of these droplets, which is sort of the green thing. And sorry, no, that's the cell. And then you express the protein. And then you take the cells, add the primers with both the chimeric primers that touch down on the gene and add the external tag containing 4 Ps, as well as the external primers. You put them into the droplets. Those are the round black things. You heat to 95 degrees centigrade, which blows the E. coli apart. And then the idea is that if whole system works, you will amplify if the polymerase is active the gene for the polymerase only if the polymerase is able to copy PZ pairs. How are we doing? You can raise your hand and ask questions, interrupting me if you would like at any point that you'd like. So that's cool. So Phil Holliger has based his career on this. So what's the problem? Well, it doesn't work usually. Now I can't do serious accusations against Phil, since he is probably the best protein engineer in the area of polymerases on the planet. But his early surfactant system, even he doesn't use it anymore. So this early water and oil system used something called SPAN80. I don't know if anybody here has ever used this. It's from animal fat, and therefore it is variable from batch to batch. And emulsions using SPAN80 proved to be unreliable. Dark precipitates formed at times at the bottom of these oil phases. Half the time the enzymes were inactivated by something that was in the SPAN80. Well, I won't bore you with all the efforts that have been made in our laboratory and around the world to clean up SPAN80. You can put mercaptoethanol in. There's some argument that there's a peroxide. I mean, it's a long discussion, but basically it has been abandoned. So synthetic materials are also problematic. So there are silicone surfactants and oils that are used in cosmetics. So for example, they were commercialized by Degussa and Evonik, and there's some companies in Japan that sell them. And every now and then you can call Evonik up and get yourself a sample of this stuff. And they actually sometimes work pretty well. But if you try to call them up a couple of years later and get some more of it, they usually don't respond to your answer because, you know, you're just, I mean, this is a billion-dollar cosmetic company and you're just one little person wanting a few milliliters of some of this stuff. So you'll discover that around the world, people hoard this material. And so we do as well. I even have some in my freezer here in case there's a fire in the laboratory, so we don't lose the seed to the fire department. But even so, compartmental stability is a problem, and you start seeing oxidation, and everybody reports cloudiness and dark precipitates upon storage. Of course, many hydrophobic surfactants are known that go with these oils. However, hydrophobic surfactants can invade the hydrophobic cores of proteins and other biologics and disrupt their function. So I mean, we teamed with Brian Pagel, who makes a career doing these things. We raised together some NIH grant money. We went into a 2-year program. A number of his papers made it look very easy, but the problem with this was, and so I usually, if I'm in the room, I use the Socratic method and see who among the students knows what the problem is and raises their hand. But everybody's being shy today. The problem is that nothing worked. I mean, and you can go back. I mean, I was preparing for this talk this week, and I went back and reviewed my emails back and forth between us. A lot of this work was done by Roberto Laus and Brian. And so, you know, we have this back and forth, which goes like, I just excerpted one of the comments. My comment at the top is, you know, we would like to be able to follow Bernath et al. And Pago's response, well, I tried Bernath okay, when I was first experimenting with SPAN, tween mineral oils to see if I could deliver magnesium in high concentrations to RNA droplets. One of your problems with these artificial cells, water in oil, is you would really like to get material into the droplet after you have formed the droplet. And so Bernath had proposed a method to do so. And of course, 49 papers cite Bernath, but nobody actually ever uses his approach. And this is a problem. But and then Brian goes on to complain about the Able EM90 system. This is Evantech, and he's perhaps about that at great length to me as well. But this is prompts what we call the student question. And the students are always want to know the answer to how is it possible for internationally famous papers and professors to get tenure and promotion, all these other things, for papers that don't work? Well, the answer to that is that science is a sociological enterprise. All the professors now have to turn off lie, speak directly to the students. You write grant applications, get jobs, get promoted by reporting what works, not by what fails, even if it only works occasionally. Okay, so now the professors can come back now that the student knows that everything we've been teaching you about science as an intellectual enterprise is all a bunch of hokum. All right, but we still desperately needed polymerases that replicate Aegis. So we turned to fluorine. Now, fluorine is an unusual atom. Fluorous species are orthogonal to both hydrophobic and hydrophilic species. They are, with respect to both biomolecules, orthogonal as well. And that means that you put a hydrophobic molecule with an enzyme, it's a good chance that the hydrophobic molecule will stick itself into the hydrophobic core of the enzyme and disrupt its function. But if you put a fluorinated molecule in, the fluorinated molecule and the enzyme usually do not interact. And so Brian actually called our attention to a nice Japanese chap working with at Dow Chemical in Kalamazoo, Michigan in the 1980s, who had reported some of these fluorinated oils. We got a hold of the guy who actually did it. He was boating on a lake in Michigan long into retirement. He did not know where the samples were. The next problem that we have is we actually want to use these fluorinated oils in cell sorters. They're generally not built for fluorinated oil, although Biorad and various other companies now are having them. And so the problem with this is that nothing really works, okay? This is the running gag. Yes, you can make fluorinated water oil emulsions, and several of these are actually widely used. However, in general, the fluorinated oils have densities higher than water. For example, HFE 7500, which maybe some of you use, commercially available, has a density of about 1.6 grams per mil. Fluorinated FC40, which maybe some of you use, also commercially available. It's used in some of these sorting devices, has a density of about 1.8 to 1.9. And so what that means is that in these media, water droplets float. Okay, so that's a problem because it concentrates droplets into small fractions at the top of the total volume. This makes the droplets difficult to manipulate in microfluidic droplets devices. They creep along the surface of the channel, they get stuck. I mean, if you concentrate the droplets at the top of a fluid volume, it makes them exposed to air. If you are exposing it to air, this allows the droplets to fuse. They also bump into each other a lot more, as if they would bump into each other if they were dispersed throughout the entire volume. That leads to droplet-droplet fusion. And of course, that destroys the compartmentalization and the Darwinism that you want for selection. By the way, Nicole Liao, who works here as well, she addressed this problem by adding cesium tungstate to the water. Now, if I were on location, I would ask if anybody have ever heard of cesium tungstate. Why that's an odd thing to add. And why did we add it? Well, the answer is there is a periodic table. I'll give extra points for anybody who can find cesium and tungsten on that periodic table. Go. Nobody wants points. Well, they're heavy, they're dense. So cesium is down at row 6 on the left, and tungstate is also over at position 74. This is about as heavy a salt as you can find on the periodic table that does not denature proteins. Tungstate turns out to be relatively innocuous, and cesium turns out to be relatively innocuous. And that's actually able to boost the density of the water. So you see our problem. The fluorinated oil has a density of 1.8. Water has a density of 1. So what do we do to keep the droplets from floating? Well, we put cesium tungstate into the water to make the water heavier so that it sinks a little bit better. Now, we never get up to 1.8. That requires a lot of cesium tungstate. But it turns out that even increasing, or actually decreasing, the buoyancy of the water, increasing its density just a little marginally, actually does help in some of these sorting devices. But that's hardly a solution. So we set out to engineer a fluorinated oil with a density of water. And it was kind of nice because right at this time, National Science Foundation came along with an eager project. I don't know if any of you know about eagers, but for the students, you should know these are very valuable because they avoid peer review. Since we're not really in this field, if we submitted a normal proposal for peer review, everybody who is in the field would trash it and we would never get funding for it. But eager lets the NSF program managers pick a project without peer review and fund it at a relatively low level. But it turns out, and we had been giving our NSF program managers lots of good results so that they could tell the good results that they had funded to their boss, who could tell them to their boss, which would increase their budget. So the NSF people gave us the commission of getting a fluorinated oil with a density of water. Now, you immediately have a problem with the periodic table, right? For those of you who can find fluorine you'll notice that it's up on the upper right-hand corner, just a little bit more dense than oxygen. And so it turns out that their intrinsic density of fluorinated molecules is higher than water. But of course, the greater atomic mass of fluorine is partially offset by the larger volume of CF2, and of course CF3. These are larger in volume than CH2 and CH3. And of course, both of those are less dense than water. And also our problem is that we're going to be using a silicone backbone. Of course, silicon has a density that's higher than oxygen and water, and so that's a problem. But we thought that we could get fluorous oils having a density of water by balancing the fluorinated components and the alkane components. And that's how this project was done. So Roberto then went back, and by the way, Roberto, who is from Peru, who has started a biotech company called Pan American Bio, which we all want you to know about, did this work, and he's made also these surfactants. Of course, the density of the surfactant is not so critical. but it is quite heavily fluorinated in this particular case. There is the structure of what he's actually made. And then there's a long story. I don't know how many of you are interested in chemistry. I could spend the rest of the hour talking about how these compounds were chosen and how their ratios were developed. But let me just give you the end of the story, which is that this fluorinated monomer, which is this three-membered ring cyclic species that's right on the top of the diagrams. We also put in the trimethyl silyl-trimethyl, which is a terminating species. So then when you do ring opening polymerization, ROP, you get a certain length under control by adjusting the ratio of the terminating thing with the cyclic thing. And of course, you see the silicon on the left has only hydrocarbons, the silicon on the right has fluorinated species. So we're adjusting the fluorinated hydrocarbon ratio by changing the ratio of those. But when you're done, you get the fluorinated monomer, 12 grams, 25.6 millimoles, and the non-fluorinated monomer, 72 grams, and the hexamethyl disiloxane, which is the terminator species. You heat it, you put a little bit of acid catalyst, you cook it for 3 hours at 75 degrees, and then you remove the unreacted, well, you decant it to get rid of the amberless catalyst, and then you distill it to get fractionations, and you end up with a remaining fluid, which is fluorinated and then has a density of about 0.99 grams per milliliter. So actually in that water with any kind of salts in it will actually sink. And so of course, what we do is to adjust the ratio of that low density fluorinated oil with higher density fluorinated oil to get the water of any salt concentration that we want to flow. So that's great. So here's some pictures. You can see what's going on here. So some density, we put these through viscosity measurements. These have actually a higher viscosity than the oils with a higher density. There's actually a trade-off. Higher the density of the oil, the lower the viscosity because it's more fluorine. It's sort of how that works. But you can see on the lower right, these are pictures of the emulsions, and they can be stable. They mix uniformly in the total volume. That dispersion is stable for weeks. You can see some droplets being formed at a T-junction. I think I have better pictures, yeah, here. So what you're looking at here is the water coming in from the bottom, going into the fluorinated oil, floating from the right and left, and forming these droplets. And you can actually see the Marangoni effect, which is the effect that arises from uneven distribution of the surfactant on the droplet. And so this is just great. So now the question becomes, what can we do in these droplets? And in particular, can we do everything that we want to do? And so it was kind of fun. So one of the things we tried was Origami, which is this using DNA from the M13 phage, which is a big circle, and then you fold that large circle into a structure. This one, it happens to be a cuboid. This is actually assembled. You can actually buy these components from a company called Tilibit. And you fold the origami by using staples that bind to one side and to the other side of this large circular M13, MP18 viral chromosome. And, we can form it. There's the scaffold on the left, about 2,000, whatever. And then the origami structures on the right, you can form them in solution. That's what's in the control sets, or that's in the control lane of this gel, or you can form them in the emulsion. That's relatively straightforward. Of course, that requires no enzymes, but PCR does require enzymes. Again, one of the reasons why you use fluorinated species is to prevent, well, if you use hydrophobic surfactants of a certain kind, they cause the polymerases to fail, and that was one of the remarks that Brian Pagel made a moment ago. But here's real-time PCR, showing PCR in bulk, that's the blue line, and in the water droplets in fluorous oil, that's the red line. So these emulsions were prepared using a microfluidic device like I just showed you. This is a microfluidic device made by casting silicon The oil has about 2% surfactants. It's pumped into this aqueous phase, which I just showed you the picture of in this buffer. And the volume in the droplets is about 30% of the total volume of the solution. So the red line, which shows you the EVA green fluorescence as this PCR proceeds, is producing at the end point of PCR amplicons just about the same as what you would expect, given that the droplets in the fluorinated oil are only about 30% of the total volume that you're observing, whereas of course the volume that you're observing in the bulk solution, the homogeneous solution, is of course 100% undergoing PCR reaction. So this shows really that the PCR works perfectly well, that the droplets are perfectly stable to PCR cycles up to the high temperature range of 90 degrees. This is not influencing the enzymes, and it's a wonderful thing, which of course is what you really need to do if you want to do certain classes of colony compartmentalized self-replication, which we've done. So it also, of course, supports TACMAN assays. And oh, by the way, I didn't make a mention here. Keep in mind that this amplicon, we're actually doing 6-letter Aegis PCR. We do have G, A, C, and T in this, but also P and Z, and there is the P and Z structure that is in the 5th and 6th letters of the genetic alphabet. So this is actually 6-letter PCR being done in an artificial genetic system capable of evolution in an artificial cell system. Here, the 2-phase with the fluorinated oil outside, a fluorinated surfactant as the membrane, quote unquote, and of course water that is inside. So it's also true with Pacman. This is actually also a 6-letter Pacman. This is one of our assays that we use to show that polymerases work. There again is Z and P, and there's Roberto who did this work, and Pan American. And so what you're doing with TACMAN, of course, is you're extending this primer. It's on the upper left of the duplex. And as it extends, if it reads through the Z in the template, you displace the downstream oligonucleotide, which contains a FfAME fluorescence molecule at the 5' end and a quencher in the middle. And there's a nuclease in the TACMAN that cleaves the fluorescence quencher from the fluorescent species. So that's how you see the signal. And again, the volume of the material undergoing active TACMAN in the droplets is less than the volume undergoing active TACMAN in the bulk. So that's why the red line, which is the real-time fluorescence signal, is lower than in the fluorescent oil than it is in the bulk. But it's about what you'd expect given the relative volumes of these two. In fact, it's a little bit better. Now, these are not droplets made by this nanostructure. We actually just made these droplets by shaking. So you take the water and the oil and the surfactant, and you mix them in a Qiagen tissue lyser. You put a metal bead in it, you do it 15 hertz for 10 seconds, and 17 hertz for 7 seconds. You have a snap cap on your bead to keep the material from being sprayed around the lab. And it's actually a perfectly good way of generating these as well. Now, the dimensions are not as homogeneous as they are with a microfluidic device, but for this type of thing, it's hardly important. But now there was another problem, and the other problem was, how do you get things in and out? So when we put triphosphates in for the PCR, or put triphosphates in for the TACMAN, they were just triphosphates. But of course, we wanted to be able to control the start of the PCR. And to do that, we went back and fell back on a nucleoside, which you are looking here, which we actually developed for next generation DNA sequencing and next generation automated enzyme assisted DNA synthesis. And that's nucleosides with the 3' OH blocked by a green NH2 group, a green amino group. These are aminoxy nucleoside triphosphates. We actually sell them for both enzyme-assisted DNA synthesis as well as next-generation sequencing. And what's nice about them is that when you have them in the mixture, they are accepted by DNA polymerases, so you will add one of them to your template. But then the polymerization stops. that ONH2 blocks further extension, so you can't do this. Now, that's fine. I mean, in that sense, it's like a dideoxynucleotide, but what's nice about the aminoxy group is that you can put in a compound like amyl nitrite, which is shown right above it, and that removes the O and, well, it breaks the O-N bond. It removes the blocking group. It removes the aminoxy. It frees up a 3' OH group, and so now polymerization can continue. So keep in mind that we have all these species also in fluorinated form, so the amyl nitrate can be made fluorinated, and it will diffuse through the fluorinated oil. Actually, both amyl nitrate will as well. And once it goes into the water, it partitions into the water and some amount. And so if you set up the droplet, which contains blocked triphosphates, the rolling circle amplification will not occur because the triphosphates, well, one base will be put on, but then the further extension will be blocked. But then you put in amyl nitrite, which diffuses through the oil to the droplets, goes partitions into the droplet, and bingo, you actually now get, right, the rolling circle initiated. And so this is what's doing, what you're seeing here. The amplification with light cycler 96 for RCA was done. Again, this is a standard RCA formation, and you're watching a number of lines. The emulsion using all natural NTPs, of course, works. No initiator is needed. The emulsion using all natural NTPs also works with the initiator added. That's just shows that the initiator is not screwing things up. But then the emulsion with the ONH2 added, you have initiator, you get a PC or RCA. But if you don't add the initiator, you don't get any RCA at all, or you get it at very long times. So this is a way in which you can get things in and out of these droplets as well, make them fluorinated. So now, okay, after all of that, we can replicate Aegis DNA. And to show you how this is done, I'll just put a few slides up that I'm sure some of you are familiar with, but we can also now make this 6-letter system into a evolvable system. It is a chemical system capable of Darwinian evolution. It is not life because it is not a self-sustaining chemical system capable of Darwinian evolution. We have to sit there and manage it. And of course, with the coronavirus likely to have been the product of engineering in a laboratory, we are very, very hesitant now to talk about creating artificial life. But never mind. This is a standard cell Celex process. It was actually, Celex is actually a trademark of Larry Gold's. Of course, Jack Shostak and Jerry Joyce then did a lot of in vitro selection following a similar strategy. The cell Celex step was developed by Wei-Hung Tan, who gave us this very nice collaborator, Li-Chin Zhang, who actually did these experiments with Kwame Sefa and Zuni Yang. It's a very simple thing. It looks just like Celex that you may have seen before, except as you can see in the upper left-hand corner, the DNA has got 6 letters in it, not 4. That's a library that's synthesized by chemistry. We actually sell the phosphoramidites for that. You then spoon the library onto target cells. These are going to be liver cancer cells. Of course, as you know, most of the components of the library will not bind to the liver cancer cells, and so they get washed away, but a few will bind. And so you extract the bound DNA, and these are the so-called survivors. You're now around 4 o'clock on this clock as you go clockwise around the scheme. Now we take counter cells. In this case, these are untransformed liver cells, and we do a counter selection where we remove from the survivors that have bound to the cancer cells, survivors that also bind to the non-cancer liver cells, and we throw those away. And now we get survivors that are specifically binding to cancer cells. And of course, you then do a 6-letter PCR with some of these enzymes that have now been coming out of CSR in these systems. And you repeat this. And after about 11 cycles, you start to get binders built out of a 6-letter genetic alphabet. By the way, with standard GACT Celex with just 4-letter DNA, these cell-targeted aptamers with nanomolar affinity take about 18 to 20 rounds of selection. So this implies that a 6-letter Aegis library is a richer reservoir of binding molecules than normal libraries. That's actually hard to quantitate, But the idea here is that nitro group functionality is sort of like a universal low affinity binder. So it's like a nitrocellulose where lots and lots of proteins will stick to it, not for any good reason. But never mind, you can see these plots. We're using now a cell sorter to see what fraction after pool 13, round 13 binds. So in the upper plot, you see the red curve on the left, that's the cell only, and then the aqua curve just to the right, this is the non-specific binding of the whole library to that cell. That library, of course, is carrying a fluorescent tag, so you can see it. But over time, you eventually get bulk binding to the cancer cells that's specific, and that's what the two panels are below. Again, the native cell, the aqua being the cells with the library at whole, and then the orangey yellow line being the ones that bind to cancer cells. And of course, they don't bind to untransformed cells, and that's because of the negative selection. And there is Z and P, which we've asked you to memorize, the structure down at the right. And it's, of course, very important now that you go back, and we have developed 6-letter sequencing technology to sequence those aptamers. There is a sequence of one of them. It's the one that binds in green. Of course, it has a single Z and a single P in it. If we mutate the Z to make a C at position 23, the green binding goes down to the aqua binding. That's a log scale, so it's about 30-fold less tight binding. And then, of course, if we mutate the P to a G, the binding goes down to the blue. And of course, if you mutate them both, the binding basically goes away. And so this is how we know that Z and P is important in the binding of that aptamer. And it's kind of fun because actually the more Z's you have, the tighter the binding. So what we do, of course, we get out of these selections with polymerases that replicate 6-letter DNA, we got a range of these things, and some of them have more P's and Z's, and some of them have less. But what I've done here is put in that table P's and Z's in red, and then rank these things based on their dissociation constants, their apparent dissociation constants binding to liver cancer cells. And the best binders contain Z and P, even though, by the way, their PCR is still the most difficult. So, and of course, the worst binders are the ones that contain no Z and P, and they're at the bottom of these. But this is just a set of aptamers that emerged from 6-letter CLEX using polymerases that are able to replicate. This is actually work that was done by Elisa Biondi, who some of you know from her astrobiology fame. Here she asked whether you could also get catalysts out of this, and the answer is yes. This is a relatively easy catalyst to do. It's the cleavage of RNA. It's kind of amusing. She gets from an Aegis library about a dozen examples of ribonucleases that cleave. Here you're looking at one of them, which has actually 4 Z's in the library portion. By the way, the black curve and the red curve in the lower left are the progress curves of this selection. Elisa is getting successful catalysts out of a 6-letter library after 10 or 11 rounds of selection. She actually never gets catalysts out of this library after 20 rounds of selections without Z and P. And the reason for this, she's using a very short library. These are libraries only about 25 nucleotides in length, and where people have gotten successful catalysis of RNA cleavage like Santoro and Joyce, they are using longer libraries. I'm not sure what the meaning of that is quantitatively, but Elisa has a new National Science Foundation project going where she's determining the landscapes of cleavage with the landscapes that show how much better libraries are with 6 letters as a reservoir for functional nucleic acids. As you may know, Irene Chen had to look at this for 4-letter alphabets and basically produced disappointing results about the landscapes, but the 6-letter landscapes look better, and Elisa has been working on that, so you'll have to ask her to come give you a talk on that material. But there's a good reason why Z might be important. I mean, in proteins, imidazole from histidine acts as a general acid and general base protein here in the cleavage of RNA. This is the mechanism for ribonuclease A, where you use 2 histidines. One is a proton donor, one is a proton acceptor. Aegis Z is also a general acid, not quite as good a one as histidinium. has a pK of about 8. But all these catalysts that we see have 2 histidine, Z's in them, which implies that they are using Z as both an acid and a base. And of course, natural DNA, GACT, is a lousy acid or base, even though we call the information content components bases. They actually don't work very well as acids or bases near neutral pH. But this may be a reason why a 6-letter alphabet is a richer source of catalysts. By the way, what's kind of amusing is, of course, the minute you build these things, you can use them. We took one of these, this is actually Li Qin Zhang again, took one of these aptamers with 6 letters that binds to a liver cancer cell, but not to normal cells. And then what you do is you attach a single strand toehold, which serves as a trigger, which then opens these 2 hairpins, which are these multicolored things in aqua in the upper right-hand corner of this picture. So these 2 hairpins, once you've used the trigger to open one of them, that generates a trigger on one of the hairpins to open a toehold on the second hairpin, which then creates a trigger that opens a toehold on the first. And so what happens is you then form a concatemer of all of these going out to the right. And you can actually see these by AFM, but the important thing here is that they bind to doxorubicin, which is a drug. And because the aptamer is specific for liver cancer cells, once it binds with the tail nanotrain carrying the drugs, whole thing is internalized and it kills the cancer cell. So this is an analogy to what are called antibody drug conjugates in modern cancer medicine. These days, if you have cancer and you're fortunate to have it of a certain kind, they will have antibodies that bind to the cancer cell. Those antibodies are covalently attached to 5 or 6 drug molecules. The antibody is then internalized into the cell, into a lysosome, which then releases those drugs and kills the cells. And what's wrong with this is, of course, to make the antibody drug conjugate requires protein chemistry where you throw reactive things on a delicate antibody. So if you add 3 or 4 drugs, it's fine, but if you add 5 or 6 drugs, you start to kill the antibody. So if you have this cancer and you're being treated with an antibody drug conjugate, your treatments are somewhere, well, some of these are like $21,000 a sitting, right? And then they strap you into a dosing chair where they put into your vein over a course of 6 hours, grams of this antibody drug conjugate. Well, with the aptamer drug conjugate, of course, we make this thing for about $200, and it, of course, carries dozens of drug molecules per aptamer. And so, but again, a note for the students, we are trying very hard to get this into the clinic, but the NIH peer reviews are still not letting us get there, even though we've gotten a very nice cancer team at the University of North Carolina actually use these. But just to tie this together, what you're looking at is an aptamer attached to a drug, which carries the drug to a liver cancer cell, where the aptamer is connected. Oops, I did not want to do that. Sorry about that. Where the aptamer is connected to bind to the cancer cells because it was evolved to do so by a selection, right? And it was evolved to do so by a selection because we had polymerases that could replicate Aegis Dna. And the reason why we have polymerases that replicate Aegis Dna is because we have the stable artificial cells able to do the technology to replicate them. So that's pretty much what I have to say. I'll stop there and be happy it's 45 minutes, yes. And I'll be happy to answer any of your questions.
Thank you. Thank you very much, Steve. There's a ton of questions in chat. I don't know, can you see chat?
I can certainly see chat. So let's go see chat. Hey, Terry to everyone, would 4 bases give an expanded genetic code so we could translate more than 20 amino acids? Excellent question. Yes, is the short answer to this question. In fact, we did that in a collaboration with a guy named Richard Chamberlain and Jim Bain at UC Irvine, and we published that paper in 1992. So yes, it turns out that we did not use the ZP pair. We hadn't invented it then. We used the SB pair. And what you can say is that with 6 letters, you have 216 codons. And so we were able to incorporate iodotyrosine was our 21st amino acid. And it turns out that the nucleic acid chemistry, once you get the nucleic acid chemistry down, the ribosome is actually quite tolerant. And so Floyd Romersberg, about 10 years later, actually showed that with these hydrophobic base pairs, you don't even have to have hydrogen bonds between the base pairs, as Floyd showed. And Floyd got extra amino acids into proteins using that. Floyd also then managed to put these into living systems and made a interleukin variant with an unnatural amino acid, started a company called Synthorx, and Synthorx was sold about 1.5 years ago to, I forget who bought it for, but for $2.4 billion. So if you think that synthetic biology in this space is useful, there you go. The catch, however, is the nucleic acid chemistry works fine, but now you're back to a different problem. How do you charge the unnatural amino acid under the tRNA that contains the funny base in the anti-codon loop so that it can read the complementary funny base in the messenger RNA? And that's a protein engineering problem. And we do not know even today, even though Francis Arnold and these people have won Nobel Prizes for doing so, we actually don't even know today how to engineer an amino acyl tRNA synthetase to take your funny amino acid and put it onto our funny tRNA. And so when we did this in 1992, what we did was we synthesized the charged DNA chemically, which means we made one protein molecule for every tRNA we put in, which is a hell of an expensive way of getting funny iodotyrosine in. So let's see, we have now from, wait a minute. Okay, let's see what I've got here. I had COVID last year. Oh, okay, I got to interview you. Let's see.
That was the answer to your question at the very beginning of the talk.
No, but when you're sharing screens and giving a talk, I don't get to look at the chat and not get distracted. Okay, could you do this polymerase selection in a liposome? Okay, that's your job, not mine. I mean, we, of course, watched Shostak suffer for years to do water, oil, water, right, droplets. And that compartmentalization is worth your life. And I mean, I'm sure Kate can tell you this as well. You've got that extra phase requires that you understand the curvature of the lipids and the droplets, and you keep calcium, and if you have an anionic surfactant, and there's all sorts of problems here. So PCR in a liposome, that's going to be for the next generation of chemists, because I will be dead by the time that gets done. Let's see, could you do the, okay, that's unfair, we're not chemists. Oh, yes, you are. Everybody in this room is a chemist, you just don't know it. Oh, okay, I see, we have a back and forth between Henry and Terry, okay. Okay, how do you do Darwinian evolution on 6-letter systems? They don't translate. Well, because we're selecting directly on, I mean, obviously, if we were trying to make 6-letter codons, that is 216 codons out of 6 letters in the alphabet, and we would need translation to do selection on the protein that's encoded by these. But we're actually putting functional groups directly into the nucleic acid. In fact, that was what motivated this project in 1985. Shostak said he was going to do laboratory in vitro evolution to get self-replicating RNA. This was a dinner in December of 1985. I still have the notes from that meeting. And that he was going to get self-replicating RNA. I said, no, there's not enough functional groups in the RNA. I mean, you need to have more letters in the genetic alphabet and then add functional groups to make that RNA look more like proteins. In fact, it sort of makes it look more like transfer RNA and ribosomal RNA, because if you go back and look at those structures, it's almost as if as the RNA world was struggling to get good catalysis, it started by adding functionality to the bases before it decided to abandon this RNA catalysis and go entirely onto a new biopolymer, proteins for which it had to invent translation. So I drew out for Jack over the dinner table all the structures we're going to make and all the side chains we're going to attach. And Jack's remark was, well, it's going to take you 10 years to synthesize these compounds and 10 years to get polymerases to replicate them. And by then we will have had self-replicating RNA. And I said, no, you won't. Now, it turns out Jack and I were both right. Jack did not get self-replicating RNA. And we took 10 years to make these damn molecules and of course, 10 years to get them. Polymerases to work them. So, okay, do Aegis base pairs pair at all with canonical? No. We published a paper in 2009 with the first author is Shuichi Hoshika, and that paper is talking about Hachimoji Dna, and because Shuichi is Japanese, and the Japanese word for 8 is Hachi, and the word in Japanese for letter is moji, as in emoji, a emotional letter. We did complete thermodynamic melting studies of all these, and we have all of the melting temperatures from matches and mismatches in these. So there is no way we have mismatches any more than we have with GACT. Now, of course, you do have them with GACT, right? So GT forms a perfectly good wobble base pair, and in some cases, if you deprotonate G or T at high pH, you will form a mismatch. And so the same thing is true with Z, for example. If you deprotonate it, it will form a Watson-Crick pair with G. And that actually is perfectly good Watson-Crick geometry. So mut-S, for example, will not recognize deprotonated GT as a mismatch. And so that's one of the major reasons why we select for polymerases, to try to create polymerases to reject charged base pairs, because the minute you start adding protons or removing protons from bases, you change their hydrogen bonding pattern and you open up all the mismatches that you can imagine based on that. Does Aegis DNA pair? Oh, sorry, that's a fascinating talk. Why, thank you. Could 6-letter genetic code create an orthogonal shadow biosphere? Okay, so I have a web page, a blog called primordialscoop.org. You should all go look at it. So Primordial Scoop, we put on Benner's random thoughts, or Elisa's random thoughts, or Jan Spacek has random thoughts. And one of them has to do with shadow biospheres. So for those of you not up to this, but shadow biosphere is a possibility that you have a life form living beneath your feet, which you don't recognize because you don't look for it in the correct way. So it's the same way as von Leeuwenhoek invented a microscope and all of a sudden discovered that there was a whole biosphere that no one knew about. Or Carl Woese, by RNA sequencing in the 1960s, discovered the entire biosphere called Archaea, which was not recognized before. So when Philippe Marlier and the team in Shanghai and the University of Illinois and in France, there was actually teams all around the world, discovered, or actually repeated, a result that was reported from Moscow State University in 1977, that there were phage infecting cyanobacter that used diaminopurine instead of adenine as their base in their molecule. Now, it doesn't change the hydrogen bonding pattern. Diaminopurine still pairs with T, but they discovered it everywhere. And in part, this is because when you go out looking for life these days in a hot spring or in a swamp or in your large intestine, what you do is you put in primers for the ribosomal genes and you do PCR amplification. Now, if that ribosome gene has a funny base and you're amplifying with polymerases that have evolved for billions of years to work with GACT, you don't amplify as efficiently G-amino-A-CT, or for that matter, G-amino-A-CT-ZP, and so you'll miss them. And of course, even if the efficiency is only a few % less after you do PCR, you basically deplete your sample of all of the interesting stuff that uses anything slightly unnatural. You also deplete of things that have, ethyl cytidine. I mean, there's all sorts of possibilities here. So the short answer to your question is yes. And it's not speculation. In some sense, that's what happened with the discovery of aminoadenine. It was just published about 2 months ago. Aegis polymers are resistant to proteases. Yes. Is it possible to expand to more bases? Well, of course, we've gone to Hachimoji, which is 8. 9 and 10 are, we just don't do because we just don't need them, but there's the 9th and the 10th is the KX pair, which we are actually using here to go into living cells. It happens to be a base pair that happens to have some value in our experiments to get this thing working in living E. coli. 11 and 12, I would have to put the structure back on, but the purine analog in J is a pathological structure right now. Actually, all those heterocycles have gone through chemical development as we have changed the structure to manage reactivity issues. So even Z without the nitro, it oxidizes too easily, it epimerizes too easily. These are things that we've had to fix. And so with J, we just haven't fixed it yet. And 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 we haven't done that just because we're still working on 8. And we still have a 9th and a 10th if we want to go further. And oh yeah, so I neglected to take Terry off. This is Mr. Dactyl, first name Terry, and he has a magnet on his bottom, and therefore he floats above a magnet in the he floats above the magnet on the and that I should have taken that off because it is distracting. But yes, that's so we work at the Foundation for Applied Molecular Evolution. If you go outside my office, there's a museum. One of the things that we have done here is resurrect ancient genes and proteins, sort of a Jurassic Park experiment. And we have fossils out there for everything that we have actually resurrected proteins from. So yes, the pterodactyl is fun. Okay, so let me just see. So this is they were asking for the paper that, there you go. So there is the paper that the link to, yes, very funny. Everyone's a comedian. Yes, so this is the link that you requested to the, this is where we worked with Jim Bain, who is the first author. And Dick Chamberlain was at UC Irvine. Chris Switzer is now actually at UC Riverside, where we used extra letters in the genetic alphabet to incorporate extra amino acids into proteins. Okay, let's see. I'm doing pretty well here. Gotten rid of a lot of the questions. Nope. Yep. Okay. Hi, Terry. You mean Mr. Dactyl. Terry to everyone. I feel seen. Yes, everyone's a comedian. Right. Did I get everybody?
I think you got through all of them.
Okay.
I had a couple of questions myself, but actually other people asked that my questions were about the translation possibilities. So,
yeah. Well, I mean, look, I mean, we had just gotten a major NIH grant to synthesize proteins which contain phosphotyrosine, phosphoserine, and acetylysine, which are 3 common amino acids in post-translationally modified proteins involved in cancer. And so, you know, you can make them by chemical synthesis, but at some point that becomes a length issue. And so they have decided that we want to manufacture those proteins for cancer standards and cancer research. And so we will do that. But the only reason we picked those 3 was because Pete Schultz, Ed Floyd, or I, and a couple of other people, Dieter Zoll at Yale, for example, Mike Jewett, Farron Isaacs, these people have managed to produce, and also some Japanese group, I should mention, already have amino acid tRNA synthetases that happen to take phosphotyrosine, a phosphoserine, and acetylysine. So we picked those solely because somebody else had solved the charging problem. Anybody who wants to solve the general charging problem should get on it if I have got a good idea, because that's really the only way in which this is going to be. Of course, then again, these artificial cells are likely to be, you know, platforms for doing directed evolution in that context, because you're going to try to evolve these things, you really do need to connect phenotype to genotype here in the amino acid tRNA synthetases. And that's going to have to be a pretty complicated system.
Yeah, I'm really excited to see something like 6 bases or 8 bases being translated and efficiently charged, because that's going to just explode the field of synthetic cells, that the amount of designs we could do.
Yeah, well, your problem is that it's not just that you have to recognize the unnatural amino acid, right? You have to be able to engineer the binding pocket of an amino adenoid, but you also then have to recognize whatever you're going to use as your recognition feature for the tRNA. Now, of course, there are a few tRNAs that don't actually look at the anticodon. So when Pete Schultz goes back to try to engineer that system, he picks those as his starting points. But in principle, you could look at the anticodon loop containing non-standard nucleotides. But the catch there is, again, the problem that's unsolved in molecular science is how to get molecule A to recognize molecule B in a condensed medium, which is water. And we don't know how to do that. We really don't know how to do that. And every time you get, we worked with Dave Baker, for example, who says he has computer programs that will design proteins. Well, every time there's no doubt in my mind that he's successful in doing bulk this and bulk that, like sort of a shape thing. But we asked him to design proteins that will take funny bases, and he has done so for us very nicely, and we've made them, and not a single one of them has worked. So part of it is not that we don't understand proteins. We don't. It's not that we don't understand ligands. We don't. But the real problem is that we do not understand water. And you got to remember that the binding of any of these things to anything is a competition between the binding of water and the competition binding to the ligand. And we don't understand water and how it binds to things. I can't tell you I cannot predict, if I didn't know, that DNA or RNA would have spines of hydration, for example.
Yeah. Right. In the interest of time, I think we have to wrap up. And so thank you so much. I really appreciate.
My pleasure.
And thanks everyone else. And see you all next week. And thank you again, Steve, for your time.